YouTube stream should be on right now. Or is it? Should be. It should be working. What? 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 No. Uh, What? Yeah. There's a lot of delay. So, like we said, I need to check the describe the required functional elements of difficult cloning vector, origin of replication, regulation of plasmid and copy number, initiation of DNA replication, selectable genes, antibiotic resistance, multiple cloning sites, number of unique recognition sites for restriction enzyme. What are the properties of restriction enzyme to form a recombinant DNA molecule? The restriction enzyme EcoR1 cuts a circular DNA molecule bearing one target sequence, resulting in a linear molecule with a single-stranded sticky ends. Because of the complementary other linear molecule, complementary other linear molecules with EcoR1 cut sticky ends can hybridize with linearized circular DNA. DNA. So, what do we learn here? Circular DNA forming a recombinant DNA molecule. To form a recombinant DNA molecule, the restriction enzyme EcoR1 cuts a circular DNA molecule bearing one target sequence, resulting in a linear molecule with single-stranded sticky ends. Because of the complementarity recognition of sequence of typical restriction enzymes. Name important properties. Recognition sequence for restriction enzymes are palindromes and therefore create sticky ends, cohesive ends. Most of the recognition sites sequence are either 6 base pairs or 4 base pairs long. The cut can either be central, creating blunt ends, or it can be staggered, creating mostly a 4 base pair single stranded end. The sticky end can be either 5' overhang or a 3' overhang. The enzymes are named after the bacterial host. H. pal was isolated from Haemophilus parainfluens EcoR1 from Escherichia coli. Hindle from Haemophilus influenza, Pistol from Provincidia stuarto. Hosts are immune against their own restriction enzyme due to DNA methylation, for instance 5-methyl C. Shortly describe gene regulation in lack of run of E. coli. You know what? Let me go this. Take this with the artificial intelligence. But I'm really scared about the exam today. I don't know if I will pass or fail. Lactopron E. coli regulates genes from lactose metabolism through a combination of negative control and positive control. Here's a concise breakdown. Key components: structural genes. LAC-Z cleaves lactose, glucose plus lactose. LAC-Y imports lactose into the cell. LAC-A detoxifies byproducts. LAC-ZYA, ZIA. Key components: regulatory elements, LACL. encodes repressor protein. Operator O, repressor binding site. Promoter RNA polymerase binding site. CAP site, cap site, binding site for cap, CAMP activator. Regulation mechanism, negative control, lack repressor. No lactose repressor binds to the operator, blocking transcription. Lactose present, allo, 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 allolactose binds repressor release operator transcription occurs. Mm-hmm. If lactose is present, it will remove the operator from the restrictor, repressor. Positive control, low glucose CAMP levels rise. Complex bind to the CAP site, enhances RNA polymerase binding. High glucose CAMP levels drop, no CAP activation, weak transcription, even if lactose is present. Lac operon is fully active when lactose is present, glucose is absent. Purpose to quickly identify E. coli colonies with successful DNA insertion in plasmid vector using visual color assay. Reported gene LAC-ZA reported gene, a truncated gene encoding the alpha fragment of the beta galactosidase, multiple cloning sites embedded with LAC-Z alpha, inserting foreign DNA disrupts the gene. Substrate XGAL turns blue when cleaved by beta galactosidase. Inducer IPTG mimics lactose, which deactivates the lac repressor to allow transcription of LAC-Z alpha. XGAL turns blue when cleaved with dactogla. No insert empty vector blue colonies. Intact LAC-ZA functional beta galactidase. Enzyme cleaves XGAL blue colonies. Successful insert white colonies. Foreign DNA disrupts LAC-ZA non-functional beta galactosidase. XGAL not cleaved colonies remain white. Ligate DNA with MCS of a vector, transform into E. coli, transform into E. coli, yeah. Plate on medium containing ampicillin, XGAL plus IPTG. Observe observe colonies. Blue empty vector, white successful insertion. Why it's useful. Rapid visual screening of recombinants, eliminates false positives, no need of PCR sequencing for initial screening. Describe main features of PCR. So blue is bad, white is good. Gesundheit. Yeah, I'm just gonna get ready for the exam. I don't care if I don't know or I know. I don't care if I fail or pass this exam because the important stuff is I know what genetics, introduction to genetic is about. So I would say I don't consider myself passing the exam or passing introduction to genetics because I still have more to understand. Peace out, YouTube. I should not have just streamed, but yeah, whatever. Peace out, YouTube.